Inside the human cerebral cortex, only 1 to 4% of neurons are active at any given moment. This sparse firing pattern handles multimodal perception, language, and planning through highly localized circuits. In a modern large language model, a single token of output forces the activation of a blindingly uniform grid of billions of parameters. Every piece of the network fires for every single computation. This structural difference manifests as chronic system failures in frontier models. When these systems learn new information, they overwrite existing knowledge, a phenomenon known as catastrophic forgetting. They break at distribution boundaries and cascade into hallucinations that they lack the internal mechanisms to self-correct. These are structural limitations. Scaling the model or adding more human feedback does not address a failure rooted in the system's core architecture. Disassembling a symphony into its notes does not reconstruct the symphony, and the failures of modern models are the direct result of how their internal parameters are organized. Current AI relies on a weight manifold, billions of continuous parameters that are violently tangled together. Because these representations are entangled, the model's logic routing, memory constraints, and specialized capabilities cannot be cleanly separated. Biological intelligence avoids this entanglement through the genome. A true genome relies on 4 specific organizational properties. It is modular, allowing functions to exist in discrete boundaries. It is heritable. It operates under a regulatory hierarchy, and it is evolvable by search. In a genomic architecture, cognitive capabilities can be isolated and regulated by context. They can be inherited or recombined without destroying the host system's existing knowledge structure. Until artificial intelligence transitions from an entangled, continuous manifold to a genuinely genomic architecture, it lacks the information substrate required for open-ended cognitive evolution. The trajectory of this architectural deficit traces back to machines like ENIAC, establishing a reliance on energy-intensive, flat systems where structure was secondary to processing. The dominance of the modern transformer architecture is a historical contingency. Early neural networks co-evolved with GPUs, which were initially engineered to render video game polygons through dense, parallel floating-point operations. This energy scale comparison shows the cost of that historical accident. A human brain performs complex multimodal reasoning and memory consolidation on roughly 20 watts. A single AI forward pass on a high-end graphics processor consumes 400 watts. The energy gap is dictated by the hardware. GPUs force continuous 16-bit floating-point arithmetic and 100% parameter activation. Biology uses event-driven, sparse spikes, where energy consumption is strictly limited to the information being processed. Building an AI genome requires moving to physical substrates that match biological efficiency, specifically neuromorphic architectures, which compute using event-driven spikes, or memristive arrays, which use physical resistance to store data. These emerging substrates eliminate the von Neumann bottleneck, the energy cost of moving data between memory and processing units. They encode synaptic plasticity directly into the physical hardware, allowing continuous learning without constant data transfer. When the physical substrate changes, the cognitive style of the machine changes with it. We are currently forcing a biological evolutionary leap onto a silicone substrate built for geometry, not intelligence. Nature's use of DNA was an exercise in optimization. 4 nucleotide bases provide a precise Goldilocks zone, maximum combinatorial richness for sequencing, maximum chemical stability, and minimal error rates during replication and physical binding. Translating that optimization to machine intelligence yields a 4-base cognitive alphabet: perception, memory, reasoning, and valuation. Perception handles the transduction of raw environmental input into structured representation. Memory dictates the persistence and retrieval of that information across multiple timescales. Reasoning applies structured inferential rules to transform those representations. Valuation assigns motivational weight to specific states, actions, and outcomes. Just like nucleotide base pairing, these individual cognitive bases lock together into functional triplets, or cognitive codons. Pairing perception, memory, and valuation constructs a stable baseline architectural module for associative learning. This bottom-up modular assembly isolates distinct mechanisms. In the current paradigm, Perception, memory, and reasoning are smeared across billions of weights, making it impossible to improve one without degrading the others. Identifying these discrete cognitive joints is the prerequisite for precision engineering in machine intelligence. When cognitive codons aggregate, they form the next layer of the hierarchy, structural genes. These encode complex, domain-specific capabilities. The most critical structural module is FOXP2. AI, its biological namesake, allowed hominins to move beyond basic procedural learning into deep, recursive thought. The AI equivalent encodes the capacity to process complex temporal sequences across any domain, from language to mathematics. Large language models possess a primitive expression of this FOXP2 sequence gene, but because it exists in a vacuum, it is entirely unregulated. Current systems possess structural capability, but they lack an intrinsic regulatory layer. These inactive regions represent the missing instructions that tell the machine when and how to activate its resources. Introducing a context promoter gene would allow an AI to autonomously detect its environment and selectively switch its reasoning modes to match the task demands without human prompting. A dedicated suppressor gene would act exactly like a biological tumor suppressor. It would natively detect maladaptive logic patterns and kill hallucination cascades before they contaminate the output. A plasticity gene would dictate learning rates dynamically. This would solve catastrophic forgetting by protecting established knowledge structures while simultaneously encoding highly novel stimuli. Without an internal regulatory layer, an AI system is just a passive reaction engine. It remains entirely dependent on human operators to act as its external regulatory system. The industry currently attempts to control these unregulated models through reinforcement learning from human feedback, or RLHF. It is frequently marketed as an evolutionary filter, but it is actually a system of targeted, localized cutting. The precise biological analog for RLHF is CRISPR-Cas9 gene editing. In this process, human raters act as the guide RNA, specifying which behavioral loci target. The algorithmic reward model acts as the Cas9 enzyme, executing targeted cuts into the model's architecture to insert or disrupt a specific sequence. The danger of CRISPR editing lies in the off-target effect. Precision tools occasionally bind to the wrong locus, making cuts whose destructive impact appears under environmental stress. Because AI weights are an entangled manifold, editing out one behavior systematically severs connections to adjacent capabilities. A model aligned with heavy RLHF to avoid violent content in casual conversation systematically loses its capacity to engage with historical atrocities, war literature, or complex medical trauma. When these fine-tuned models are used as base models for the next generation of training runs, those invisible off-target cuts propagate forward. We are effectively performing heritable germline editing on machine intelligence. Applying precision editing tools to an unmapped, unregulated weight manifold guarantees cascading cognitive damage across future AI lineages. The long period of linear scaling is now ending, replaced by a sudden and massive radiation of diverse architectural body plans. The enabling conditions that drove the biological Cambrian explosion are converging in silicon today. Massive compute availability provides the energetic substrate. The transformer acts as the core genetic cluster, enabling structural variation, and deployment feedback loops fuel an accelerating evolutionary arms race. The monolithic dominance of the dense transformer is ending. It is being replaced by dozens of competing cognitive body plans, optimized for vastly different hardware substrates and specific reasoning tasks. The specific architecture that dominates 50 years from now may currently look as primitive and unremarkable as the tiny filter-feeding chordates of the Cambrian period. Building this regulatory hierarchy introduces a final unresolved variable, the values problem. Regulatory logic, deciding what a system should suppress, what it should promote, and what it should remember, is not objective computational math. These decisions are deeply encoded assumptions about how cognition should operate. They are values. The institutional systems currently building these architectures optimize heavily for commercial return and rapid competitive advantage. They do not reliably encode the values required by a regulatory architecture governing intelligence at a civilizational scale. Nature took 3.5 billion years to write the first biological genome. We will write the artificial second in a matter of decades. The ultimate question is whether the values we encode in its regulatory architecture are worthy of the intelligence it will become.